Hi, welcome. I hope you're doing well. Today's video is going to be dissecting a really basic question of how does protein affect insulin resistance. And I'd actually initially picked out this study because it was a bit contrary to a lot of the research that I've been looking at in relation to saturated fat. So not only is this study going to be looking at protein, but it's also going to look at saturated fat. So I will discuss the saturated fat aspect as well, but I am probably going to spin this more towards the protein metric or measure. For anyone that is interested in voting, I have a community post at the moment that I just released a few days ago, wherein I'm asking for your opinion if you specifically would be interested in me teaching a course on how to read research studies. So a little bit like what I'm doing here, but it would be far more well edited, thought out, a lot of information related to different techniques, like different experimental techniques that researchers use, how to read graphs, and then how to understand statistics, and then, of course, for me to explain how I go about, when I first open a study. Obviously, it can be incredibly overwhelming when you first look at all the graphics and whatnot, and obviously the language itself. So what are the first things that I attack to get a rough idea of what the study's on, and then how do I delve in deeper into the study design and how the studies ends up working out. So if you're interested in that, actually, if you're interested or not interested, either way, it would be a big help if you jump over to the community post. I'll see if I can link it because I would really love to have some form of indication on interest. Obviously, this is a way that I can empower individuals and kind of cut through. So it's a highway, a fast track through university education. So you don't have to, so generally all this kind of stuff that I'd be teaching you is stuff that you'd be learning in a university, and probably not in undergrad, not much in undergrad. You'd be learning it in grad school and beyond. So this is it's an advanced teaching. So I'm teaching a lot of advanced things, but I don't want it to be just for people that already have a background. The idea is to just pick someone off the street that doesn't have a background in science, and for them to be able to then follow these kind of stepwise instructions and be able to read really any paper. If any medical paper, physiology, if that's like physiology, if that's nutrition, if that's cell biology, molecular biology, all of that would be encompassed in this course. So for the details, like I said, check out the community post. I go into a little bit more detail on it, but I don't want to prattle on about it in this video introduction. But if you're not familiar with who I am, obviously I just went over how to read studies and whatnot, so you might be thinking, well, why in the world would you be qualified? I love, what I've been, something that's been really making me chuckle of late, which is related to what I was just about to say. Why are you qualified? I've had a lot of people call me like kid in the comments, and or once you get out of your early 20s, that's what some people have been saying. Look, folks, I'm well into my 30s. I'm not a kid. So, I guess maybe I look younger than I am. And I do have some experience behind me. I'm not saying that I have decades of experience or anything, but I spend every waking moment that I have that I'm not doing actual lab work, like actually pipetting and stuff like that, or spending time with friends. I'm reading research on the weekends. That's what I do. So I read a lot of studies, way more than what I present on Physionic. So you know, I'm just saying all this to signify that I have more experience than just the years, but also the years aren't, it's not like I'm 23 or anything like that. I have a few years under my belt at this point. But anyway, who am I? If you're not familiar with who I am, my name's Nicholas Verhoeven. I'm a doctoral candidate. I'll probably be graduating, estimate, I'd say like a year and a half. I'm well through, done with all my courses, and I've passed my oral exams and all that stuff. So right now it's just getting a paper out and doing presentations and stuff like that, kind of fine-tuning my dissertation project. So I'm a doctoral candidate. I've been doing cell biology research for a number of years now. I don't know, I guess with my master's and undergrad included, probably like 6, 7, 8 years, something like that. And that's where I'll leave it. Let's go ahead and jump into this paper now that I've, like I said, talked for long enough. And we'll get potentially some answers. There's some kind of disappointing results when it comes to this study, but we'll get to it. All right, so let's flip on over here. So the question that we're answering, and there's an additional question, does protein impact insulin resistance? Insulin resistance being the sensitivity of our body to insulin being secreted into your bloodstream, that insulin then binding to your cells, and then the cells then accepting blood sugar, so reducing blood sugar. That's important to keep in mind because of the metric that we're going to be looking at. And the other question that we'll be kind of peripherally discussing real quick, because like I said, this was initially supposed to be for my investigations into saturated fat, which again, this study has different conclusions than what I've found across multiple studies. So I thought it'd be interesting to cover. And that is to look at saturated fat and its impact on insulin resistance. So the study we're going to be investigating today is called Diets High in Protein or Saturated Fat Do Not, intriguing, affect insulin sensitivity or plasma concentrations of lipids, which are like triglycerides, fatty acids, lipoproteins like your LDLs, HDLs, VLDLs in overweight and obese adults. Okay, but we're going to see if that's actually the case, because my conclusion is actually slightly different from what they have in this title here, but not by much. All right, so let me remove myself real quick so you can actually see this. This is, I'm just distilling here the study design. So they had 159 participants, men and women, all overweight, so a BMI of at least 25 or higher. I realized that, as always, people hear BMI and they immediately want to dismiss things. But when you're talking about the average person, BMI can be pretty useful. So all these individuals were overweight, and they were put on a 4-week baseline diet. Now that I think about it, I should have written that in my notes as to what that baseline diet was, but I'm sure it included probably lower amounts of protein, a decent amount of fat, and probably moderate carbohydrates. That would be my guess if you're going to create a baseline diet. And the reason why they do that is because they want to make sure that all these individuals come from different backgrounds, clearly. And all these individuals are going to be on different diets or have consuming on a regular basis different diets. So they want to put them on a baseline diet to kind of wash out the effects of all their previous diets, all their previous nutritions, and then they're all on the same page when they start out. And 4 weeks is definitely a good amount of time. Sometimes researchers only do like 1 week, which is usually enough as well. But 4 weeks, good to go. Then after those 4 weeks, they're split into these 5 different conditions. So the control diet, and I've got it listed here. The C is for carbohydrates, P is for protein, and F is for fats. So, and then they had different proportions of saturated fat. So we've got our high protein, which is right here. 30% of their diet was protein, and high saturated fat, meaning that the fat level was 35%, but the saturated fat was 15%. Now they had the exact same situation, but a low saturated fat, so everything else is the same. carbohydrates 35%, protein still 30%, fats are still 35%. When I mean fats, I mean total fats. And then now, because it's a low saturated fat, it's 7% saturated. And then they've got a moderate protein, which drops the protein from 30% down to 20%, and it cranks up the total fat to 45%, but it maintains saturated fat at 15%, and then a low saturated fat condition of the same thing at 7%. So I like how they designed this. This is pretty well thought out. And then they ended up being on all these diets for 4 weeks. So split, I think it's like 30-something participants, as we'll see in the next slide, for 4 weeks on each diet. All right, so now, as always, I get that, you see this and this is really overwhelming, but don't worry. This is going to be pretty short, pretty easy to get through. This is all the different measures that they looked at. We're not going to be looking at all of them, and I'll tell you why, because only one of them changed. Throughout the entire 4 weeks, only one measure changed. So if you look at weight, nothing changed. BMI, obviously nothing changed. Body fat percentage, nothing changed. And how we read, well, I'll explain how we read this in just a minute, but waist circumference, and then like insulin sensitivity, the acute insulin response, AIR. You've got the, I think this is for MCRI, I think is the removal of insulin. So how quickly can you clear insulin, clearance rate. Triglycerides, total cholesterol, LDL cholesterol, HDL cholesterol, none of those changed for any of the diets. Okay, so at least by the comparisons that they're going to be doing. So let's hone in on the one thing that actually changed right here. Okay, so usually I don't add graphics and stuff to these long form videos because it just takes too much time. But in this case, I had to because it's just entirely too confusing to go through and try to make sense of everything. I'm not a huge fan of the way that certain studies show their statistical significance, as in like indicating a difference or an effect of a particular treatment, like a diet, like this. So A, B, B, C, A, B, C. That to me is just, it's really like, it takes me forever to try to figure out. And imagine, well, let me explain this real quick, and then I'll say, imagine having to do this for all of them. So what we're doing here is, and I've got this color coded, and just so that it's easy to identify the differences. So any one of these where there isn't the same letter, so AB is going to be the same as, is not going to be statistically different from, let's say, AB right here, because they're the same. But if you have any difference, then that means there is a difference. So AB would be different from C because C is different from AB. It's like, it makes sense, like in a simplistic standpoint, it makes sense, like, okay, it's the letter is not included in AB, therefore it's different. Okay, fair enough. But you try to do that every single time across so many different conditions. To me, it's just, it's really cluttered. So anyway, this orange line indicates that there's a difference between the moderate protein, low saturated fat, and the control diet. On the other hand, then we look at this, so BC, we compare that against A because there's no BC, or B or C here. So then we got the comparison of the blue line, then the green line. So again, A is compared to C, and then AB is compared to C because, again, those are all different. Now imagine doing all of that for all of these. That would be, I mean, it's doable, but good gracious, it's just, I just don't like it. I really like asterisks, and they sometimes use crosses and different things like that because it's easier to identify. For me, in my head, it's easier to identify. If you disagree, let me know. I'd be interested in how, like, maybe your brain works, but for me, this is more difficult. Regardless, I'm done ranting. All right, so how do we read this? We read this in that here we've got what they've done is they've taken the first measurement. So they took a measurement. Let's go back here. They took a measurement at the end of the baseline diet. And then they took another measurement at the end of the 4 weeks of each one of these diets, right? And then they compare the difference between those. So let's say let me go ahead and cut to, so you can actually see my hands. So here we've got the baseline diet. Here we've got the control diet. Or let's do something else. Let's do the high protein, high saturated fat diet. And this is a point. So this is an average and this is an average. They get a result for one and they get a result for the other one. So if one of them increases, let's say blood glucose increases, blood sugar increases in the high protein, high saturated fat diet, then they're comparing the difference between those two. So this one might be, let's say 1 and this one is 2, then, well, 2 minus 1 is 1. So now they have a calculated difference between those two. That's what they're showing here. Okay, so they're showing the calculated differences right here between the baseline diet at the end of the baseline diet, or right before they went on the control diet, to the end of the control diet. This was the difference. This was the change in blood glucose levels, meaning that there was a 0.07 millimole per liter increase. You don't need to know millimole per liter, but just for accuracy to have the units. So this is the average of all the participants that were in the control diet. So 31 participants were in the control diet. And then here they did the same thing. It's the exact same thing. It's just the baseline diet at the end of the baseline diet versus the end of the high protein, high saturated fat condition. And there were 32 individuals. So you see that there's a decrease of 0.17 millimoles per liter. Okay. And then you just do it similarly for all of them. So it's not like we're comparing the end of the high protein, high saturated fat diet, like the value itself, versus the high protein, low saturated fat diet value itself. We're comparing the actual change. All right. So hopefully that makes sense. Maybe you don't care. That's fine. But it's just to explain things. So it's also to explain that you could have you can have negative numbers, as in you see a reduction in this measure, a reduction in glucose or blood sugar levels. Or if there's no minus, then that means there's an increase in the blood sugar levels. But of course, that doesn't mean it's statistically significant. So this value next to it means that there's a range of where the true value could be. So statistically, it's been calculated to be, let's take the control diet, 0.07 increase in the average, or in in blood sugar levels. So that's the increase in the blood sugar levels. But is that the true value? Is that, like, in the real world, is that what you would actually see? Well, there's a certain level of uncertainty, and that level of uncertainty gets displayed in something known as a standard error of the mean, or a confidence interval. And that, I realize I'm explaining a lot here, and that then allows the true value, not just the 0.07, but the true value could be actually 0.08. And the statistics are just picking a number that is probably the most accurate, and then the standard error of the mean is giving you an indication of some error level that could be appropriate. Now, so the point is that we can't necessarily just look at this and then say, well, that's increasing, this is decreasing, therefore there has to be a difference. No, because statistically there's not, because there's a large standard error of the mean. There's a large error, area that between the comparisons. So even though these two look different, they're not actually statistically different. Okay, so that was a lot of explanation. Maybe that wasn't necessary. I don't know. The point is, sometimes I like to throw things out there so that people who are interested want to know. Anyway, so here we see that there's a decrease in the moderate protein, low saturated fat diet compared to the control diet. Now that is partially due because of this actual decrease, and that is partially due because of this increase. All right, fair enough. The next one is that high protein condition, again, that was what, 30%? Yeah, 30% protein. The high saturated fat diet had a decrease in glucose and had an increase in the low saturated fat diet. So they're going two opposite directions, leading to a statistical difference between the two. And then the next one is high protein, low saturated fat versus moderate protein, low saturated fat. The moderate protein shows this decrease, and obviously there's a slight increase. Again, if you were to compare this to the control, although this increased more than the control, it's not statistically significant. But because this increases and the other one decreases, it creates enough of a difference that it's statistically significant. Hopefully I'm getting that across clearly. And finally, moderate protein and high saturated fat, if you compare that to the moderate protein, low saturated fat, the low saturated fat condition shows a reduction in blood sugar levels. Okay, so what does this tell us? Well, it's not just this, but they showed some associative data. So they showed some correlation data as well. Let me go ahead and go back here. So they showed some correlation data as well, where they mentioned BCAA levels, so branched chain amino acid levels. And they wanted to correlate the amount of branched chain amino acid levels to a lot of these different metrics. So looking at insulin sensitivity and things like that. And they found a weak correlation or a weak association between increasing amino acid levels and decreasing insulin sensitivity, meaning that the higher the protein levels within the bloodstream were, as measured by BCAAs, branched chain amino acids, which are components of proteins, the lower the insulin sensitivity. So there was an inverse relationship, meaning higher amino acids from proteins, lower insulin sensitivity. That, however, is an association. That doesn't actually show you a cause of, yes, this is the cause of that insulin sensitivity. It just means that they're running in parallel to one another. You know, there's some sort of link that could be between the two. But here, going back to the actual causative to the actual addition of a treatment of high protein and actually then measuring the insulin sensitivity and things like that. Well, let me go back here. We see that this is a measure of insulin sensitivity, this SI measure right here. And we see that there's no huge differences, so no huge changes. So, the control, there's a slight increase in insulin sensitivity, but it's not statistically significant, so it could just be random chance that that's the case. You see a slight decrease with the high saturated fat, high protein. You see a slight decrease with the high protein, low saturated. It's just so on and so forth. So all of these, however, don't end up being statistically significant. So what does that tell us? Well, it's really hard to say. All we're seeing is decreases or changes in blood sugar levels. That's all we're seeing. But in other measures of insulin sensitivity, we see no changes. So in blood sugar levels, it does seem that with a higher protein diet, you tend to have higher blood sugar levels, except in this high saturated fat condition, which is counterintuitive to all the other data that we've looked at on Physionic and the other stuff that I have to show you yet. But on the other hand, the best outcome was the moderate protein, or let's just say the lower protein than high protein, and low saturated fat condition had the greatest reduction in glucose. Again, these comparisons aren't pre versus post. They are compared against, the different diets. So this ultimately leads to a very simple conclusion that we can get to right here. There's weak evidence, and I cannot stress this enough, that the evidence is incredibly weak, but it is there. I don't want to discount it. It is there. That points to elevated protein consumption increasing blood sugar levels. But there's zero evidence, no causative evidence that protein actually has a causative effect in increasing insulin resistance. Because all the other measures that we looked at, there were no differences. And the other thing that I wanted to add, for the main reason that I picked up this paper, was saturated fat. There's no indication that saturated fat led to worsening insulin resistance or decreased insulin sensitivity. They're both the same thing. So what's going on there? Because that is completely counterintuitive to the other research that we've looked at, the multiple other studies that we've looked at. And I think, I'm just speculating, but I think it's not the time. I don't think it's the 4 weeks, although that could be a factor because some of the other studies that we looked at were months of time. But I would say that's probably secondary to this other reason that I don't think their saturated fat condition was high enough. Their high saturated fat was 15%, which, according to some recommendations, you should aim for between 10 or 12% or lower of saturated fat. So increasing it by 3% for 4 weeks just isn't a huge increase. Most of the studies that we've looked at up to now have looked at 20, even 30% saturated fat in the diet. That led to those led to pretty robust effects. So I certainly don't want to discount the data here, but there's only one really weak piece of evidence, and it's related to blood sugar and the direct measures of insulin, direct measures of acute insulin release, measures of insulin sensitivity. None of them changed. So that's where we have it. I'm certainly going to investigate this protein aspect more because the researchers of this study did point out that multiple studies show that protein can be beneficial for insulin sensitivity. And guess what? Multiple other studies show that proteins can have a negative effect on insulin sensitivity. And they speculate that it may be the type of protein that a person consumes. So that is something we will have to investigate into the future. For now, I think that this is really weak evidence. If you want to take it and run with it, go for it. I would not. All right. But hopefully you learned something from this. Hopefully statistics or me just rambling, whatever it was. And I'll hope to speak with you in the next one. Have a good one, guys. Bye.